Okay, so I'm Jitendra Sharma. Yes. Let me start with one. um, so basically, you wanted to say something?
No, I just wanted to say to our guests, Ganesh and Seth, uh, anytime, I mean, you are, of course, welcome to be here for the entire duration, but at the same time, if that feels too far afield from your interest, feel free to.
Will not be offended.
If I'm bored you enough, you can. Should we start?
Yeah,
Okay.
yeah.
So glad to be here, and I'll give you a general overview of the brain development, which involves a series of events, beginning from initial layout of synapses, the brain cells of different kinds and other things, and their specialization, their localization in specific areas of the brain, and their elements of the circuits, which are like axons, dendrites and dendritic arbors, and then synapses and all sorts of connections and other things. And then once, so this slide I took from a talk from Beth Stevens a couple of days back actually at Broad, and I thought like this actually captures the essence of what is going on. Like early in the birth, during the birth, just after conception, the neural tube starts to form, and there are these synapses and different kinds of neurons. They start to sort of generate, and then there's a precocious growth for up to almost 6 years. When there's an exuberant sort of growth of all circuit elements, number of neurons which are born, and their connections start to establish everywhere. And then around the same time while this is happening, there's also some sort of synaptic pruning that starts concomitantly. But the pruning goes on till about like late in the adolescence period, when the circuit starts to fully mature, and it stabilizes and sort of it captures the map of the world in which we live. That's one way that people looked at it. So just to recap, what happens, I should do it here also. So while our focus is on synaptic pruning, but I thought like it would be a good idea to just recap what happens early in the development. So just after conception, a blastocyst forms, and then there's a neural tube formation happens. This period of first like 3 to 4 weeks, it is called neurulation period, in which neural tube forms and starts to fold. Then there's a process of elongation and cell generation, then cell migration, and then comes the period of like proliferation, where patterning happens, the synaptic sort of synaptogenesis and the interplay of the molecules of neurotransmitters as well as proteins, etc. starts to happen. And at the same time, some of the neurons start to die under some influence of retinal waves, as well as other intrinsic sort of influences which are not fully known. And then further maturation and specificance starts with the activity dependent mechanisms, and other cells which are like glial cells of different kind, microglia, as well as astrocytes and all. They are the support cells. They start to create a scaffold to stabilize the system in the circuit elements, and they also are sort of housekeepers, as well as they seem to also participate in sort of synaptic transmission. Okay.
So, Jiten, does neuronal death begin to happen within the womb?
No, afterwards.
After,
Yeah.
Okay.
Post gestation period. But it is still unclear. Like people have looked at only the mouse brains for this, and I think our understanding is still
Hmm.
incomplete. It may be happening earlier also.
I see. And one other question. So is there any kind of linkage in time of the sensory front ends and the cortical areas that are going to be responding to those sensory front ends? So for instance, in vision, is it the case that the primary visual cortex develops before the eye is fully formed, or does the cortical development follow the sensory front end development?
I think the cortical, the front end happens first.
First, I
Yes.
see. So like the hair cells and the retinal
Retinal
ganglion
ganglion
cells
cells will come first. Yes, yes.
I see.
But there is a considerable overlap. Like in the next slide, there's a timeline. So after neurulation, like between week 4 to week 12, there's a neural proliferation when the cell elements have already formed, most of them. And then this process of migration, synaptogenesis, as well as aerialization, all these things sort of happen in a correlated fashion.
Your question was before conception, right?
Before,
Or was it
before
after?
birth.
Before birth.
Before birth,
Before
sorry,
birth.
before
Right.
birth.
Before
Yeah.
conception would be remarkable.
Sorry,
Yeah,
before birth.
before birth.
Yeah,
Right.
before birth.
So like younger animals, smaller like rodents will, many of these things happen after birth. Whereas in human being, like first 10 days of say maturation is very, in mouse it is much faster, for example. By the time a mouse is born, it is very early on, but almost all of the synaptogenesis and other things are taking place slightly later. But this is the timeline for the human system. So basically, there was an important paper in 1997 from this Hattenlocker and Arun Dabolkar. They look very systematically at cadaveric brains of human brains at different time points, okay, to sort of see where, like, right from in utero brains to early developmental phase and up to adolescence. And they also did the same thing in macaque brain and try to compare how it is happening. And basically, their paper says it's a really very nice paper. So, and very highly cited paper. So synaptogenesis in humans, it begins around week 20 to 27. Okay, and that is in utero still. And it occurs concurrently with the dendritic and axonal growth. Okay. So everything is happening at the same time, literally. And initially, there's an increase in synaptic density also, so the like proliferative growth, and which is more rapid in sensory and motor cortices. Okay, and it reaches maximum around 3 months postnatal. And on the other hand, like in frontal gyruses and frontal cortex, it continues to increase up to 15 months. Okay, and the elimination is it is slightly controlled. This paper says that elimination starts slightly later. Initially, there's a synaptogenesis, and synapse elimination starts a little later. Like first, the maximum sort of density stabilizes, and after that, the pruning starts. But there are other papers which show that there's some sort of overlap. So I'm not sure what how hard this evidence is. Especially in humans, it is difficult to know.
I see. So that large amount of synapses, so the high degree of synaptogenesis, is that kind of indiscriminate?
It's
So
indiscriminate. Yeah.
indiscriminate.
Yeah.
So just neurons are wiring up.
They are developing, they are generating, they are wiring up, and they are sort of aerializing at different locations and all. And then they're sort of aerial identity also is created at the same time.
Isn't it the case that there's some migration in adults?
Migration in adults. It's a good question.
I think there was a report about stem cells developing and migrating to the proper position in some part of the brain in adults.
I've not, maybe I've not looked at that, yeah.
In the dentate gyrus, like,
What?
because that's been important, I guess.
Yeah, so
in the
so
dentate
that
gyrus,
is,
the olfactory system.
yeah, so new neurons like are generated there, but I don't know whether there's a stem cell based growth happening there.
You mean stem cell based growth or from progenitors?
What's that?
Do you mean from stem cells or from progenitors?
Yes,
From progenitors?
yes.
Oh, yeah, progenitors.
I've not seen, but I've also not looked at that. So it's an interesting, yeah. Like elimination also, like it starts and there are periods of like, it may be happening continuously, but there are rapid periods where a lot of elimination is taking place. That happens early on. It begins around year 3. And again, it happens earlier in the sensory and motor cortices, and it ends around year 12, 11 or 12, where in the prefrontal cortex, it starts later and it stays later. So it almost ends around late adolescence period. Okay. And there are implications of that. There's some of the, like the schizophrenia is now linked to that aberrant synaptic elimination later in the adolescence period, which is a precursor to schizophrenia. So many young adults are diagnosed with schizophrenia around 18, 19, 20 years of age, but they have aberrant sort of synaptic elimination during year 11 to 11, 14, to year 14. So that may be causative, but why it happens, it is still sort of unclear. But again, like the evidence is slightly, is more on functional MRI based changes and things. I don't know how hard the evidence is. So yeah.
Yeah, you had the question. Oh, sorry.
Yeah. Go ahead.
I just, if you have any insights into like the progression of myelination also.
So yeah, so myelination is in the previous slide.
Because I
was showing,
first see
the
it's
myelination.
throughout adulthood, and there's a
Yeah,
few
it's happening around.
bursts.
It starts a little later because the axonal growth has to happen first, and the myelination happens later. So that has been shown to start something like in some other place I've read about week 24, the myelination starts, and it continues till adulthood.
Yeah. I'm a little bit interested in myelination, so if you have any knowledge about I guess, the progression of myelination and how it relates to, I guess, the functional wiring of visual system or anything.
Yeah, I can give you, like, there's a bunch of literature on that. Yeah.
I've seen some of it, but
yeah. And it is still sort of especially the genetic influence of the myelination is an active area of research actually. So basically, at birth, we have about 100 billion neurons that make close to about 1 trillion connections, and this happens at about year 3. By adulthood, we lose about 15% of neurons and the corresponding amount of other circuit elements like synapses and other things. So I was talking to Jeff Lickman. Actually, he was in. There's a new sort of grant which has been. I'm also part of that. But for brain connectivity, it's a large sort of multi-PI project, about 130 million. And we are basically we are looking at 4 different species, right, from mice to marmosets, macaques, and human beings. And we are trying to make the whole cell atlas of connectivity of the entire brain. So he in his talk he was showing, Jeff Lickman is the granddad of brain connectivity right now, and he quoted this that our brains start out wiring for every possible contingency basically, okay, because we are the brain is in a new world, doesn't know about, so it starts to connect everywhere. And then as over time, it starts to prune itself to sort of map the world around us, how to navigate, how we live in this world and other things. So all those things happen a little later. So if we change the world, we may have to change our brains first.
But then does that happen the same way for everyone?
Like unless there is some genetic sort of aberration somewhere.
How do evolutionary narratives fit into that?
You should,
So-
yeah. So yesterday there was a talk from Nenad Sistan. He's at Yale. Yeah, he has come up with very insightful. His talk was like unbelievable. Yeah, you should talk to or see some of his papers. It's very informative. Yeah, Nenad Sistan, he's at Yale. So he is looking at brain connectivity and brain development and evolutionary influences, right from the genes to proteins as well as other connections.
This talk was at the Broad.
His talk was abroad, yeah, 2 days back. Yeah.
Email.
Need to get an admin.
Yeah, he's a,
Environmental conditions have an effect on the pruning process.
yes, it does. And that can, so like right now, a lot of work is being done on, there are some very nice papers that are emerging in which like chronic stress can also do synaptic pruning. So synapse loss happens because of chronic stress also. And it has been like shown that in prefrontal cortex especially, there is a synapse loss in people with major depressive disorders and all. So chronic stress does that. And it seems that the role of inhibition, especially GABA, has been implicated majorly.
Like that is implicated in every disorder.
Yeah, exactly. So basically, yeah, either you control excitation or control inhibition. But there was another, so we always thought that there's a balance of excitation inhibition, which is the key player. But there was a paper from Wong Riley. She's one of the very early persons who was with Carla Schatz, and they have some very nice papers on retinogeniculate sort of synaptic proliferation and all. And there was a review article of hers in somewhere, I think, Journal of Computer Neurology or somewhere, where they have found that actually in critical period time, there is the balance of excitation inhibition is disturbed actually. There's more, so it is more inhibition as compared to excitation, and that is necessary for the critical plasticity. Yeah, they have some evidence of that. I don't know how many people have followed it, but it was an interesting something that was new, which was because we always think that yes. The activity and activity independent mechanism as well as even the internal mechanisms are sort of trying to balance excitation and inhibition. That's why the Hebbian plasticity and everything happens. But it seems that during the critical period specifically, this balance is not a necessity. Actually, it is the other way around. So I can send you that paper if you
Any of these?
Yeah.
Yeah. So myelin and the OGM.
So yeah, okay, just to give you a little bit more about, because I have been reading a lot more about some of the genes that are in the synaptic elements and all. So I just thought that I may give you a little bit insight into this, that this is a very, very complex structure. Synapse is not just some sort of a shape, a bell shape or something like that. It's chock full of different types of proteins and other elements. So if on the top this is the electron micrograph of on top left is a small electron micrograph of a synapse. You can see there's a presynaptic and postsynaptic site on the top, and then there's a synaptic junctions that are in red. if you can see. And if you look at a canonical circuit, which is like everywhere, basically, say pyramidal neurons, extrated neurons, they send their axons, which synapse onto the dendrites of, say, a recipient pyramidal neuron. But at the same time, they also synapse onto an inhibitory interneuron, which sends its own axon to the soma of this recipient neuron. So this is a circuit which basically balances the excitation and inhibition. Okay. So the same cells that are synapsing their axons on the dendrites of a recipient side, they also synapse onto an inhibitory interneuron. Yeah. So this is from a very nice paper from Tom Suthoff. Tom Suthoff he won a Nobel Prize with Randy Shackman for defining these circuit elements and the vesicles. So yeah, it's a very, very nice sort of review, which is very readable.
So the soma of the pyramidal neuron has synapses?
Yes.
Huh.
Yeah.
So what is the actual interplay of excitation and inhibition here? Like, is there some timing delay, or does the inhibition from the interneuron arrive at the same time?
Inhibition with the interneuron, which is dependent on the excitation from the axons of the two neurons.
How about Renshaw cells? You have connections to the cell body of the axon.
Yeah, exosomatic connections also.
How is the synapse elimination is triggered?
So that's a loaded question, actually. Activity-dependent mechanisms are one, certainly, that less active synapses, which are getting much less information, they start to weaken, and they are sort of tagged for elimination. So, like Carla Schad says, there's a lecture by her which is on the YouTube. You hear that she said, like, it's like a rose bush. If you want very good roses, then you have to prune it. Okay, so to provide that strength of development to the leftover sort of healthier sort of bush elements, and prune away the things which are unnecessary. So that's one way that probably is happening here also. So use it or lose it kind of thing.
So I guess, are there exceptions to this? Like, are there any neurons that don't sort of wire in such a way where they have an overabundance of connections and then they prune? Like, are there, I'm thinking maybe like there's some specialized like innate circuits that might wire in a different way, or is this these strategies?
This is a general strategy, but specificity is slightly less known. But it is true that this actually further along I have a so this is like basically three types of refinement that takes place. So if on the top there's a refinement of the topography. So it's a single axon which is sort of proliferating everywhere. Okay, but then some of its branches are eliminated, and they are in the right place. Topographically, they are in the right place, but there are too many. So some of this will be eliminated. There are mechanisms that people have proposed, but there's no hard evidence which one are tagged for sort of elimination. Is it that the centroid of this thing, of this location, is the determinant for the elimination of others which are sort of outside that centroid? That is one sort of theory that people have proposed. There was another paper in which people have modeled that the one that reaches the fastest the centroid of this location, which is already neuronally specified because of some afferent genes, they basically start to push away the others. okay, because this is my territory. Now others go away. And then they start to weaken and they fall apart. The other one in which there's a convergence, like multiple elements of, they converge onto the same soma. And over time, like only one will survive, the others will fall away. And that starts to proliferate within. So initially they are all sort of scrunching together in the same small space. And when the ones that are not used die away, the leftover one is, they start to proliferate. So there's a two-stage process of this. And the other one is in which, so this is also another mechanism in which the axon starts to proliferate both in the soma as well as in the dendritic arbors. Okay, but then the ones in the soma, they get eliminated. So this is from a textbook. They did not cite the actual paper in which this has been shown. So it seems to be true, but I want to see the hard evidence in a paper.
Which textbook? Which textbook?
I can send you that. It was on circuit elements, textbook circuit elements. I was looking at some things in which I can show that this is what is happening in different times.
And are there more types? We're just seeing three of them, or is it established that there are three?
These are some of the types. Again, like if I go back, I jumped a few slides. I want to show you some other things that are important. So at least important to my work right now. So this is again like Tom Subdah's paper in which, so basic idea is that synapse is a very complex sort of structure where it's a macromolecular structure in some ways. So there are multiple genes and protein elements of each of those genes. And there's a pre and post synaptic side, but it is sort of communicating both ways. It is not like one-way communication. And there are hub elements, which are neurexins, which are very important compounds. So what you see is on the also postsynaptic side, there is this is a postsynaptic density membrane. So it is just underneath the postsynaptic membrane. There is a thick layer of proteins. About 1,000 proteins are here. Okay, and the proteins that are marked with asterisks, they have been implicated in some neurodevelopmental disorders. There are almost 130 or 140 different neurodevelopmental disorders which are implicated, in which these proteins are implicated in some ways. And one of those is the subject of my research, which is SHANK3 proteins, which the loss of that protein, basically, the children, those who have this gene abnormality, they have a syndromic form of autism, which is called Phelan-Bechdel syndrome. So children which are born with this, they have profound hypotonia, and they have intellectual disability, and about 80 to 85% of them also have autism. So this is a monogenic form of autism, like MECP2 is Rett syndrome, that's one, and then FMR1 is one another monogenic form, and the SHANK3s. So this is the interest of this gene is that this gene is in the PSD, or the postsynaptic density. So it is a synapse formation gene. Okay. It's a complex sort of structure, and it communicates with other sort of protein elements here. And there are multiple isoforms of that, but this gene, when it is absent, there's a high percentage of children will have intellectual disability and autism.
And the gene is expressed all over the brain.
So yes, but there are loci of high expression, especially in the striatum and in hippocampus as well as frontal cortex. Yeah, so again, like so this is the SHANK3 gene complex, basically. All about this different circuit elements. So you can see like a single gene element is so complex. It's chock full of different connections and different molecules. So coming back to sort of synaptic pruning, there are several questions, but they are only partially answered. One is how synaptic pruning happens, like what are the causative elements of that? Why some synapses are lost? So there's some evidence, of course, the activity-dependent mechanisms and all, but no other sort of hard evidence is there why else it happens. Thank you. Then, of course, how many synapses will be eliminated and at what point in time, and why others get reinforced. Of course, activity-dependent mechanism again. But before activity starts, there are spontaneous retinal waves also that which reinforce some of the synapses, and the others are dying down. Why it is selecting something, and the reasons for that are not known. And again, like role of neural activity in retaining, why something is retained and why others are pruned. It is not fully sort of set in stone in some ways. And of course, now the newest research in the elements of immune system, the microglia, they appear to be essential in carrying out the elimination process. So it starts on like early in the development, the microglia does that. And it continues throughout, even in adulthood. So the basic idea is that pruning on ORI in children and teenagers is increasingly recognized as a causal to the multiple psychiatric disorders, especially.
What's the signaling system between the microglia and the synapse?
Yeah, so like usual, these cytokines have been implicated. IL-7 and IL-27, these are the two cytokines that seem to trigger microglia processing to start sort of engulfing the synaptic elements. But it's an inflammatory process, but why cytokines are specifically targeting some elements and not others? Is it that the synapse is already weakened because of lack of activity, and then it starts to sort of get tagged there because weakness means there's some sort of a endogenous inflammation, and that needs to be sort of pruned. So yeah, Beth Stevens has done some bunch of work on this.
So are you saying these, synapses are seen as a, detrimental, as a health issue for the brain? That's why the immune system attacks those.
Probably, or it is one way of housekeeping itself. If you have too many residents in a small room, okay, you have to take out some so that others can function properly.
So you're thinking of Amir's limitations, where when we're talking about zeroing out some of the blank spaces in the receptor fields, that's kind of like eliminating the weak connections.
Yeah.
That's what I remember actually. But I'm just curious if there's any chemical change that causes, that triggers this mechanism, immune system, to start attacking those synapses. I don't know. Is there any findings, for example, that establishes that there's physiological change, or there's a chemical change, or maybe bonds are changing? I don't know, the proteins, for example.
So if you read some of the articles from Takao Hansch, he has done some of this. So he has used GABA agonists and antagonists to sort of change the critical period earlier or later, increasing or decreasing GABA. That can sort of slide this critical period earlier or later. And you can even extend the period by manipulating or modulating this. So he has used benzodiazepines as well as other GABA agonists to do that. So that is one. And then he's also shown some, maybe somewhere I have a slide. There's a enzyme activity is called TPA. That seems to play a very important role in, as well as BDNF. It's a growth element. They both. So if you do a monocular deprivation for a long time, and then you locally put BDNF, you can recover the lost axons and the synapses. So yeah, so growth factors with sort of fibrin components as well as the enzymes which are in the synapses, they all play a role.
I have a question. I think Rani once mentioned kind of the relationship to memory. So if all this learning is happening, how does it affect information that we collected during the younger years? Because it's quite some years that there's a lot of learning. So is that affecting?
Yeah, so in adulthood, like for example, now people have shown, say, Alzheimer's and other things, that there's a synapse loss. Okay. And that has been linked to dementia as well as loss of memory and other things. So like what is causal to that is still a question. But it is certainly true that there's a huge synapse loss. We probably lose about 10% synapses every 10 years after 30 years of age.
Yeah. But during those early years,
Mhm.
I mean, if a baby has experience in year one
Mhm.
and created some memory,
Mhm.
is there kind of a guarantee that those
memories
are
will-
going to be pruned, or is it because there's so much pruning, it's not really guaranteed?
So again, like I think there is there are some models which propose that there is a capacity for memory, okay, and those memories which are not used often, they start to fade away. So the child may be having tons of memories, but he or she may not use those memories later on in the period. And there are some more important which are used for survival or for whatever they are doing. So they get strengthened, and they are, but it's again like slightly speculative. And whether they can be ever recalled or not, or they are tagged in such a way that they can be quickly recalled, but with a much more reduced sort of brain efficiency.
Is the pruning, I mean, to what extent does it connect to biological advantage? If one, if a child would grow up in an environment that was very dense with vegetation as compared to one that was nearly devoid of vegetation, is there pruning and an optimization of a
Yes.
certain,
Yes.
in this evidence?
Yes, there is an evidence. So there was that famous sort of study on Polish children who were kept in a very sort of secluded environment in which they were not allowed to go anywhere and to interact with anybody. And they seem to have some of them when they died and they looked at their brains, they had much fewer brain connections. Okay, because of the deprived environment in which they grew up.
And it
So
could go the other way.
It can go other way around. But again, like if there has to be some sort of a limit imposed by the environment in some ways. Okay, you cannot, like, if I keep giving you input every day, a new input, will you continue to proliferate more synapses to retain that? Again, like it's a question which is not fully answerable. But they do say that, like, when a child is growing, the more varied experiences you give to that child, the more sort of intellectually able that child is.
So does the evidence even go into the realm of perceptual ability in terms of nuance, perception, visual perception that would maybe translate into thought capacity?
Yes, I'll show you some evidence for that. And like it's a indirect evidence, but let me So where do I? Okay. So basically, in postnatal period, like the retinal, when you look at the retinal geniculate system, okay, this is from ferrets, cats, and mice. There's a proliferative growth which does not which goes which does not follow any sort of pattern for specific eyes. Okay, and then under spontaneous waves that Carla Schatz had shown that they start to prune and they start to have their areal identity. So left and right eyes, they start to segregate into layers.
What's P10?
Post-birth.
Post-birth, yeah. So P birth is P0 and P10. So first 10 days of birth.
10 days.
Yeah. So postnatal. And then postnatal 10, this spontaneous wave have been recorded by multiple people. And around the same time, this the segregation of left and right side inputs in the LGN that starts to take place. And then after the pruning, within the same area, there will be more synaptic remodeling. Okay, the more stronger synapses they remain, the other ones which are less used. So this is the activity dependent mechanism. So initial postnatal development is all proliferative growth, then endogenous spontaneous wave, they start to sort of specify left and right eye layers, and then further refinement happens with the activity dependent mechanism. So these are the three sort of developmental mechanisms which people have studied hugely, and there's a lot of literature on that.
In the primate brain, given that in the mature brain there are these beautiful segregations of the ipsi and contra eyes, is it the case that prior to birth the layers have a mix of
layer
ipsi and
yeah. So when the human child is born, or even a primate, yes, yeah, yeah. So that's why, like the Hubel and Wiesel's thing, that it is the critical period is the one where all this thing is happening. But when a primate is born, it is already there. Okay, so something in utero is happening, which is doing this.
Would you say that if a child has more experiences and then they have more synapses form, and then as a consequence more pruning, do they have a higher chance of abnormal pruning, or is there no connection?
That's a good question, and certainly, like, so excessive pruning has been implicated in some of these disorders which develop late, say schizophrenia and all, and less pruning has been implicated in autism, for example. So early on, there's less pruning happening up to first 3 years of age. So there are too many synapses, and that may be one of the reasons that autism starts. So, but then again, like this a spectrum. And so I've already discussed this part. So basically, in the sum total of this retinal geniculate pathway is that if you block the spontaneous activity, which Carla Schatz and other people have done, in both eyes during early segregation period, they fail to prune the retinal movements. Okay, so there'll be a mishmash of this thing. If there's no, if you take away all the spontaneous waves.
So, can you still remove normal eye input? So, like input related activity? And
so that happens later. Right now, it's like the spontaneous waves are before, in early birth, before eye opening, when the external input. Yeah,
that's
exactly.
What you're saying,
yeah,
a lot of the stuff
exactly. Yeah. But again, like this also happens in utero in, say, primates and in humans, okay? Because the retinal wave period is already over by in the first 10 days. Yeah. And then where is the activity coming from, which is sort of segregating the channels, and it is not fully known.
The spontaneous activity is, people mainly think it's the retinal waves.
Yeah. Exactly. That's why if you put a sodium channel blocker, then you can abolish all retinal waves, and then there will be there won't be any segregation.
So the retinal waves between the two eyes are correlated?
They are correlated, yes. Yeah. So if you uncorrelate that, If you close one eye, then you can see the deprived eye starts to lose it. It synapses faster.
Do we know how to generate how the retinal waves are generated? And by correlated, you mean they're like synchronous?
Yeah,
They're
synchronous.
synchronous.
Yes,
Yeah.
synchronous.
And do we know how they're generated?
So there are molecular mechanisms that people have investigated. If you so Carla Schatz's papers and many of her papers are they've done so epibetadine based signaling was another one mechanism that they had initially used, in which they had this is a molecule that they use in the sort of nasal side versus the temporal side right now, and they could see a differential change there. So there are multiple molecules that are implicated.
But I guess how do they synchronize across the eye?
A good question. I think it is not fully understood, or maybe I've not read them to retain them.
And these are like they're like a few times a minute the waves.
Yeah, so it seems like in mice it is every 15 seconds or something like this. There's the wave generated. Yeah, there's a temporal spread of that. But again, like so, retinal waves require a synchronicity, whereas between the two eyes, whereas the activity-dependent mechanism they require unsynchronous input from the two eyes. Yeah, if the two are synchronized, there is sort of ocularity change. Yeah. So eye-specific segregation and synaptic refinement is dependent on asynchronous activity from the two eyes. So, yeah.
That kind of like I guess distinguishes the patterns from the two eyes.
Yeah. So one problem with this whole sort of field and literature is that everybody has their favorite area of work, and their reviews and all their papers only talk about that. Like Carla Schatz will only talk about retinogenetic rate system and retinal waves and all, and she has a lifetime of her work. Even if you listen to her talks, like 80% of the talk it won't be on retinal waves and other things. And then you see Takao Hansch's paper and all. So he will only talk about like this critical period and how it can be changed with the inhibition. Okay. So everybody has their favorite thing, and they don't sort of do full justice to the entire field fully. So that has been an issue.
That means that there's room.
There's a lot of room to, yeah, exactly. Like for example, earlier I did not know what it is. Once I did the rewiring work, I got clarity on that most of the retinal inputs, they don't go to LGN. About 80% of the input is going to supracolliculus. It's only 20% which is here. No, in cats and ferrets also. The major input is going to the colliculus.
What's the proportion in primates?
Primates
a lot.
Yeah.
Really?
Yeah.
80% in primates.
Probably 60 to 70%. Yes, but the major input is still on this collicular side. Yeah, and
You were thinking of getting rid of your colliculus.
exactly.
Okay. So, yeah. Okay.
Yeah. So, but again, like there is some sort of high eye specific segregation there also in the dorsal layer of supracolliculus. But how hard people have looked at, there were old papers of Dr. Sharma and all that they had used. They had looked at that carefully, but after that, not much work has been done. And again, like deprivation, what does it do? Does it follow the same rules as the geniculate or not? That sounds
Yeah, in mice.
So no. Go ahead.
Well, I mean, there's all the literature on the corticate mice. You remove the entire cortex from the mice. They just like walk around like everything is fine.
Yeah, exactly.
They still see things.
So why do you need cortex?
Etc.
Exactly.
They don't do like two things, which I think is like groom themselves and like trim their nails. That was exactly what they said. Yeah.
Yeah. So yeah, so basically, early on, Google and even people before them, they had done this eye injections in one eye, and they'd seen that there's a ocular dominance columns are there, and like this is all textbook stuff, and um, there's a ocularity change. Like most of the cells are binocular, whereas some cells, about 20% of them are monocular. So ocularity still persists, but there's general sort of spread, which is like a Gaussian. And if you deprive one eye, for example, as human and weasel did, you can shift the ocularity to the open eye. And if you do both eyes are closed, then this is some other people have shown a slightly different sort of, there's an acuity loss, and generally there's more binocular cells. Okay, so monocularity is reduced to an extent, but also there's a spatial frequency change, and the contrast also is changed. So acuity loss is there with this thing, and spatial frequency as well as the contrast.
Have people talked about what are the factors underlying acuity loss?
Again, like people talk only of the activity lack of activity that improves the visual acuity as well as contrast.
This is of relevance to Prakash.
Yeah,
because,
exactly.
Great. And one of the open questions there is, so we're assuming that much of the effect is coming from changes in V1, but even at the level of the retina, given that the ganglion cells pooling fields likely are affected by the lack of pattern stimulation at the level of the retina. But we don't know whether the eye is providing these high spatial frequencies to the brain, or is it the case that right at the level of the retina or at the level of the ganglion cells, you've already cut down on high spatial frequencies. So the brain is only receiving low spatial frequencies.
Exactly. But then given all that plasticity to an extent is retained, if you do a hyperstimulation of, say, high frequencies, high spatial frequencies, can it be
right? So I think that's exactly the interesting question. So if things have happened at the level of the retina, so the retina is simply unable to signal high spatial frequencies, then no matter what we do to the brain, the front end is now got that. But if we find that the retina is still capable of signaling high spatial frequencies, and it's the brain that is lacking, say, sensitivity to these high spatial frequencies, then there's some cause for hope that you can potentiate the response to high spatial.
So in binocular deprivation, it is not that all cells have lost their contrast sensitivity. It is reduced, but that doesn't mean that it is not retained. So like there's a sharp fall off in the spatial frequency also, but that doesn't mean it is zero.
Right. Yeah.
So that gives an idea that there's a residual sort of contrast sensitivity and spatial frequency sensitivity which is still there, and there's a way that it can be sort of improved, if not significant.
Yeah. I think that's a fascinating possibility.
Like that happens with even in like motor cortex and all. Like in mice, which are you if you remove part of the motor cortex, of course initially they are sort of impaired, and then you put them on these wheels and other things, they start to gain much faster. So hyperstimulation does have some. But again, like, should it be selective or not selective?
Right. So sorry, just to pick up on that one.
Yeah.
So in one of the graphics that you showed with monocular deprivation, where the balance so the upper panel there. So the number of cells that are visually driven, the total number is still the same as in the normal brain, or do some cells just become completely
Completely
unresponsive?
unresponsive.
They do.
Yes,
I
yeah.
see.
And the same thing happens with the binocular deprivation. There are more cells which will lose their sort of activity.
I see.
Yeah. Yeah. So there is a, I think it is about molecular deprivation. You lose something like 10% or so, but in binocular deprivation is about 20 to 22%. Cells will go.
Are there any fMRI-based markers that can act as proxies of total number of cells that are visually sensitive? So, say in a highly simplified world, if the average brain of a sighted person shows a certain level of response in V1, and we see a lower level of response, say in the Prakash kid's brain, would we legitimately be allowed to say that there's lower sensitivity or fewer cells that are visually selective?
I don't think it is too sort of blunt.
Too coarse, yeah.
Too coarse. But certainly one can do a diffusion imaging, and you can actually get a sort of a fractional anisotropy thing and see whether they're more myelinated axonal fibers going there, and then they should be correlated with how many neurons are getting reduced. So that may be one. So I can tell you this in my rewired ferret work. Some of the residual like rewired ferrets, I had done diffusion imaging with Vanvidine. And at least in some couple of ferrets, we did see that the axonal fibers are going towards the from LGM, they were going towards coursing towards the visual cortex, and they were turning back towards auditory cortex. The only big problem was that because of the lesion that I made, the brain also sort of became distorted.
Right.
So whether that sort of turning back was functional or it was because of the distortion of the brain was difficult to understand or sort of justify. So that's where we stopped at. But, so again, like it also, at least in primates, there is a age-dependent deprivation change in the sense that if you do it in critical period, the functional and structural changes are much more profound. And if you do them much later in life, deprivation happens, say, 14 or 15 years down the road, its effect is much less.
So what is the y-axis?
So the y-axis is basically non-deprived eyes, occupies like most of the layer 4, and the non-deprived
what proportion of cells is responding to?
Exactly.
Okay.
Exactly. So on the left side is the anatomy, and the right side is the function. And so, but this has been like the newer papers are sort of refuting that, but all of those are done in mice. So again, like the jury is out whether it is happening in the primate. So this is the primate work.
I see.
So strabismus, again, like the quality of input or type of input also has some effect. If you cut the rectus muscle, then of course you're distorting the vision, and that's, you can see that there's a total loss of binocular vision. And then of course people have done this. They raise the kittens with the goggles, with vertical goggles, and even strabismus, like with the strobe light and all. So in strobe light, there's a sort of the direction sensitivity losses. Yeah.
I once actually asked about that, the like vertical goggles or whatever, and the development of nystagmus. Why are you laughing?
So is your question, where can we get these vertical goggles? Party classes.
Yeah. And I guess the development of nystagmus.
Yeah. So it's a good question, but certainly even with people who have open eyes but they have lost vision, there is a profound strabismus.
Oh,
But what is the use of that? It's like that your system is trying to get any input from anywhere by moving the eye much more, like
What do you mean,
involuntarily.
steps?
so does the amount of nystagmus increase with like length that you wear, the length 85 or something, like the amplitude or whatever?
Good question. I've not seen any papers on that. But maybe I don't know whether people have taken a hard look at whether the cat is having nystagmus or not. But I do know that if you train monkeys with ratings of different orientations and all, the vertical orientation creates nystagmus quite a bit, and you have to sort of habituate the animal to keep changing the spatial frequencies of the gratings and all. And finally, they start to fix it better.
Very high frequency gratings probably generate more nystagmus.
I did not look for that, but what I did was like I used different spatial frequencies to habituate the animal to less nystagmus, and temporal frequency also. So its temporal frequency had much more effect, like faster going gratings, like large wide gratings faster. They had more profound nystagmus.
The temporal frequency of the instance.
Yeah. Because probably the eye was trying to follow
It's like a
the
drum.
The chronic use of the prism, would there be a neural adjustment?
Yes. Actually, Shigeru Tanaka had done this experiment in monkeys.
Nature.
Yeah. It was in Nature, 1995 Nature. Because he had come here to give a talk at that time. So he put these dove prisms to switch the eyes on either side.
And then he was getting activation in the IPSI.
IPSI side mode. Yeah. And but the monkeys were very, very sick. They had like profound sort of amnesia and a lot of other problems, because the world was distorted for them so much that they could not navigate.
Amnesia.
Yeah.
Wearing.
They would be just not sleep, and they had gastric issues and other things.
Just from chronically wearing.
Yeah.
And you don't adapt to it, like you might.
So he claimed that at some point in time they were adaptation, and that's when he did all the studies. But
Okay.
so it was a very like profound study at that time. So again, like I was telling you about the short-term deprivation, the synapses are not lost, but their motility is changed. Okay, so probably their visual acuity and sensitivity will be affected with short-term monocular deprivation. If you do it for 4 days, then there's a total synapse loss, and the axons also start to retract. Okay. But then if you do a long-term deprivation, of course, and also apply locally this BDNF, you can recover the lost synapses. So this is Takao-Henshin's. So this pruning also occurs in other parts of the brain, like there's this climbing fibers in the cerebellum. They also have this thing that they start initially synapsing onto the soma, and then they start to translocate. And the ones which are staying with the soma, they die off. And the ones that are translocating to the dendritic side, they start to strengthen.
Is that what they're called, climbing?
They're climbing because they're climbing from one place to another. Yeah, so cerebellum also has this. Then, of course, I told you about this microglia thing. And there's a lot of interest in that because neuroinflammation has become a big deal. And people are thinking that besides the proteins and molecules, it is the the inflammatory responses, which will be more important than anything. So again, like too much and too little, both ways of pruning can be deleterious. For example, in schizophrenia, there's an excessive pruning, whereas in autism, there's much less pruning. But they are at different temporal sort of windows.
When you say altered circuits, do you mean like some of the connections are not supposed to be pruned, but they're pruned? And therefore the configuration of the circuit
Probably, but another way is that somehow they are tagged for pruning for some reason, and so they are pruned by the system. Why? Nobody knows. So there are two questions here. One is that they should not have been, but they are. But the other way, the other side of the coin is that they are somehow tagged for elimination, and they are tagged.
I see. So the tagging might be an error.
Yes. So or sort of activity of the tagging mechanism. For example, so like cytokines, if they're in an overdrive, as like the people, that was one of the mechanisms for issues with the COVID also. There was an avalanche of cytokines. And that's why people have all sorts of issues with the cardiovascular system and all those things.
Mm-hmm.
So that may be a causative mechanism. Mm-hmm. So like just to recap that synaptic pruning helps efficiency. There are some papers on that, more on modeling rather than actual hard sort of evidence. Then specializing that, of course, the activity-dependent mechanisms are supporting that part. And again, use it or lose this. And strengthening of the synapses with LTP or LTD, that can be another mechanism which has been implicated. And environmental factors, as I told you, like my own work on rewiring the cortex. So the basic idea was that we asked this question that if what is the effect of the input on specialization of the sensory cortices? Okay, so if I put the connections of the eye, the retinal connections, to go into the auditory area where they are not supposed to be going somehow, what will happen to the auditory cortex? So we did this experiment in which on the day of birth of ferrets, so when ferret pup is born, their retinal ganglion axons are almost reaching the thalamic sides. Okay, so they have not reached the cortex. Okay, so it is the time that you can reroute them. So what we did is we basically most of the connections go to supracollicular, so we cut that supracollicular part, and we also created space in the auditory thalamus by taking away inputs from the inferior colliculus, which is coming from the cochlea and all. Okay, so now it is the auditory cortex was not getting the input that it normally gets, and it is now getting aberrant inputs from the retina. And the question was, will it become visual or will it just reject all the? So we recorded from single neurons from, say, a normal ferret, and they were only just selective neurons, which we expected, of course. And when we did the same thing in rewired auditory cortex, there were neurons which were also orientation selective. Okay. But then the next question was, do we have a topographic map of orientation selectivity as you see it in sort of a normal ferret? So this is a normal ferret. You can see that if you showed gratings of different orientation, you can get these maps of orientation preference, and there's a beautiful sort of topographic map with the single domains as well as the pinwheel structures where all orientations meet. So what happens with the rewired cortex? So on the left side is the normal V1 of the same ferret. Okay, because we spared the LGN to V1 connectivity. But since supracolliculus was severed, so those connections started to go to auditory cortex, and we had the same orientation preference map. And then the question at that time when we submitted this paper, the reviewer asked is, how do you know that orientation preference map is actually the functional map? Like, are the neurons really there which are showing the same orientation preference? So I did this experiment again in which we recorded from neurons, put electrodes in the orientation domains to see whether the same orientation preference is there in the single neurons or not. So we saw that indeed it was true. But as you can see that this map is not as clean as the map here in the normal visual cortex. So there is something, the intrinsic mechanisms are there, which are sort of hurting a smooth mapping or fine-tuned mapping. But certainly neurons were there, which were. And then after that, like we also wanted to see whether there are structural changes or not. So in primary normal visual cortex, if you put the clusters of similarly liking orientations, they are connected together for long ranges. So I put an anatomical label in one of the domains and looked at normal V1. You can see that there are clusters of orientation preference maps, which is in the same line, for example. But if you do in a normal auditory cortex, because it is a unidimensional, like the sound is a frequency axis, so you can only see sort of cells which are connecting to the same sort of frequency axis. But then when I did the same thing in photoid cortex, it was more like a V1. So there were patches of orientation selective neurons which were all over the place. So this was like the input activity. Basically it has an instructive role on specifying what the cortex will be. So what is visual about visual cortex? Because it is receiving the visual input. But there, of course, there are other mechanisms that constrain that. So yeah, there are some papers, like there were Alison Barth at UPenn. She has some modeling papers that you should read. Yeah, so different brain regions will have different critical periods. So the heterochronicity basically. So during the synaptic pruning is most active. So they have shown that is a necessity, at least in their modeling work, that it helps in more robust and more efficient networks. And of course, several developmental disorders have been implicated in this excessive pruning or too little pruning. So yeah, but again, like we lose about 10% of our synapses after 20 years, every 10 years. So, but that doesn't mean that we lose everything by 80 or 90 years. There's a lot more sort of neural sort of real estate there. But there's some very interesting papers on this chronic stress, which are very reliable, where there's a synapse loss, certainly, and it can be reversed. Like recently, the role of ketamine has been for the depressive disorders, where there's a regeneration of synapses with greater inhibition. So yeah, this is the study I was telling you about, the amblyopia. So they are very close to doing a clinical trial. They are doing a short-term sort of putting the TTX in the eyes to stop all activity, and then so you uncover a hyper or meta activity after a quiescent period, and that seems to sort of regenerate the plasticity in the visual cortex. So these are some of the references I can give you. You already have this, so yeah. So basically, yeah, this is the summary. Yeah, so it is still an enigma, like why pathological processes do synaptic pruning, and how does it happen? Because there's certainly a developmental pruning which is time controlled, but then why pathological processes also do scientific pruning, and how does it happen?
Thank you. This was
Yeah.
really.
I hope I was able to give you something which is useful.
Neurons have to be in the right place though, don't they?
Yes, they have to be in the right place. If they are not in the right,
Dystopias are no good for this.
yeah, there is no, yeah, they have to be in the right place at the right time also.
That's right.
Otherwise, they will be rejected.
Otherwise you get seizures.
So, in nature also, this is the same thing, right? So in marmosets, sometimes triplets or quadruplets are born. And since the mother cannot nurse more than two at the same time, the weaker ones are sort of left over by it. So there's some sort of a natural selection. That the healthier one is the one because they have a stronger chance of survival. The mother feeds them more. And the same thing must be happening here also, that somehow something gets weak and then it is sort of rejected by the system.
It's a huge organizing principle in nature and evolution, etc.
Yeah.
I mean, people have described it, right? Neural Darwinism, isn't that?
Yeah, neurodevelopment.
Gentlemen.
Maybe, yeah.
Yeah. But so like there's no place for weak people.
Like even in a nest, the early
Yeah.
hatchlings push out the other eggs.
The other
that
ones,
don't
yeah,
hatch.
exactly.